Hey everybody, Miss Jotzi here again. I think it's about time that we had a quiz. It's been so long. So we're going to have a quiz on Tuesday this week on DNA structure and replication. So I just want to do a real quick review with you. I really hope that you've been keeping up and doing all that you can do. I know these are desperate times, and sometimes you're just going to have to take it upon yourself to figure out what you don't know and watch some videos and search the internet and, I don't know, read your book or something crazy like that. Okay, so first up, things that you need to know for the quiz. There's going to be a lot of questions about the structure of DNA, so these are hopefully the easy ones. I hope that you know that DNA is made up of really long repeating units called nucleotides, and nucleotides have three parts. The first part is the sugar, and it is called deoxyribose. The second part is the phosphate, and that's the phosphorus molecule surrounded by a bunch of oxygens. And the third part is going to be one of the bases. Now, sometimes they call them nitrogen bases because obviously nitrogen is going to be an atom found within them as well. So remember that it's a pentose sugar. Gosh, can I draw that? Oh, I kind of did. Well, it's not the worst thing. So here's your deoxyribose. It's going to be connected to your phosphate. Put a little D here, and then the bases are connected here, and bases are found in two different kinds of forms. Some of them have double ring structures, some of them have single. You don't need to know that much. What you do need to know about bases is that the bases are complementary, meaning that certain bases tend to fit together simply based on their electron bonding. So A will often and should bond with T, and in DNA, G will bond with C. So a possible question I could ask you is, let's say that you know that there's in a total in the nucleus of any particular cell in your body, you have 10% adenine. Based on these rules and based on the fact that for every A there should be a T, that means that there should be 10% thymine as well. Add those up and you get 20%, which gives us then 80% remaining to split up equally between our two remaining bases. So that should tell us that there's about 40% C and 40% G. So that was mentioned in the notes and I think in the video. So I don't think it's tricky math, but I would just want to make sure that you can know how to do that. There'll be an example one like that on there. On the quiz. There's also going to be some questions about identifying some of the scientists who contributed to the model of DNA, particularly the model though. So there's the always two most common ones, Watson and Crick, who were partners. We've got Rosalind Franklin and her partner Maurice Wilkins, which were kind of responsible for figuring out that model that Watson and Crick first worked on were not so great. And then they had that photo 51, which helped prove that it was a helix with the bases in the middle. There's one other guy, Erwin Chargaff. I think he's mentioned in our notes, but I didn't mention him in class. Erwin Chargaff came up with this idea, that he proposed that pretty much for every, there tends to be equal amounts of A's and T's, and there tends to be equal amounts of G's and C's. So he was another person responsible for figuring out the base pairing rules. Okay, so in addition then to structure, there's going to be some questions about replication. So hopefully you can identify a picture of replication when you see one. You're also going to need to know some of the enzymes, which I just reviewed in the previous video. So functions of things like helicase and DNA polymerase, and DNA ligase is on the list. Then we have topoisomerase, one of my favorites. I'm not going to ask you about every single one because that would be a very long and tedious quiz, but just to understand the general idea of some of the functions of some of these guys. It's a pretty short quiz, and I hope you do very well on it, but I will be grading it soon, and I'm just about getting ready to put some grades into Skyward for all the work you've been working on the past couple weeks. All right, keep checking Skyward and make sure you're keeping up with your studies. See you soon.